Hello, my dear friends. Let us talk about PCR. Polymerase chain reaction is a DNA amplification process where a million copies of the same genes are being produced from a target gene using specific sets of reagent and reaction conditions. This experiment consists of the basic polymerization steps like denaturation of the template DNA and annealing of short DNA sequence, which acts as the primers for the DNA polymerase to extend the newly formed template following base complementary with the template DNA. All the steps takes place in a single reaction tube subjected to changing the temperature and time. PCR has been used for a wide variety of purposes like molecular cloning, gene expression analysis, mutational studies, as well as the genotyping. This particular technique has become an integral part of almost all molecular biology experiments that are being conducted in different laboratories around the world. Now let us see about the principle of PCR. The PCR method is based on the DNA replication process being carried out in a repetitive manner. By changing the reaction conditions at each step of the competitive cycles, it requires two single-stranded DNA primers to designate the two ends of a target DNA. From these primer sites, the tagged DNA polymerase starts adding the free nucleotide that is being added in the reaction mix following the general DNA base pair complementation principle till it reaches the end of the target gene. Once a single copy of the PCR product has been generated in a particular step, it functions as a template for the second cycle of PCR. At the end of every cycle, the amount of the target DNA is doubled. This process is then repeated again and again in every cycle of the PCR reaction process until millions of copies of the same gene are produced. In any PCR reaction, the researcher has to make a reaction mix of several components. All the components that are required in a PCR reaction mix, along with their functions, we will discuss later. After all the components are mixed, added to the mixture, the mix is then incubated in a thermal cycling process in a PCR machine known as the thermocycler. Now let us talk about the PCR components and their functions. First is the tagged DNA polymerase. Thermostable DNA polymerase functions to synthesize new strands of DNA based on the template strand. Second is the reaction buffer. Maintains the salt concentration pH of the optimal activity of the tagged DNA polymerase. Third is the forward primer. Binds to the 3' to 5' bottom strand of the template DNA required by the tagged DNA polymerase as a cue to mark the end of the chosen template. Fourth is the reverse primer. Binds to the 5' to 3' top strand of the DNA template required by the tagged DNA polymerase as a cue to mark the end of the chosen template. Fifth is the DNTPs, a mixture of all ATP, GTP, TTP, and CTP added by the tagged DNA polymerase during elongation of the primer ends, following base pair complementation with the process with the template DNA. Next one is the template DNA. The template DNA is being amplified during the PCR reaction. Next is the nuclease-free water to maintain the optimal concentration of each of the reaction components. Guys, for more videos, like, share and subscribe your own YouTube channel, and further detailing of the PCR we will discuss in our next video. Thank you.